archetype cell, and a relationship was established where that cell persisted inside the host archaeobacterial cell, and the proteobacteria, the aerobic bacteria, became permanent residents inside that cell. And then eventually established was this obligate intracellular relationship between the proteobacteria and the host archaeobacterial cell. And through time, it's suggested that the aerobic bacteria existing inside that archetype cell became mitochondria. And the same line of evidence goes for the chloroplast as well, being derived from cyanobacteria. That's called the endosymbiotic hypothesis, or endosymbiotic theory. And so we see this evidence that the mitochondria is related to aerobic proteobacteria, and the chloroplast is related to cyanobacteria. And different things support that. Mitochondria have their own DNA that's closely related to the DNA of alpha proteobacteria. Same for the chloroplasts. They relate to cyanobacteria. They have their own DNA. Those organelles, the mitochondria and chloroplasts, have their own ribosomes, basically the same size as ribosomes found in prokaryotic organisms, both aerobic bacteria and cyanobacteria. They replicate inside the cell like bacteria would replicate. They are surrounded by two membranes, an inner membrane and an outer membrane. Chloroplasts have a thylakoid inner membrane, just like the thylakoid membrane found in prokaryotic cells. So there's enough supporting evidence to make us realize that long ago, probably 2 billion years ago, archetype cell ingested alpha proteobacteria and eventually chloroplasts, which were cyanobacteria, and a permanent relationship was established, and that's called the endosymbiotic theory. Now, with our cells, the typical eukaryotic cells. Now just know that there is no typical cell out there. There are all kinds of different cells, from amoebas, the paramecia ciliates, flagellated cells like dinoflagellates and euglenas. There's all kinds of different varieties, thousands and thousands of different cells out there. We look at two typical cells generally in introductory courses. A typical animal cell, like a cell you might scrape from the inside of your cheek, and a typical plant cell. And so let's run through some of the basic components of these cells. So this being our typical animal cell. First and foremost, a typical animal cell is surrounded by plasma membrane. The membrane that surrounds a cell, all membranes that surround cells, are basically made up of the primary constituent called the phospholipids, and it occurs in a phospholipid bilayer. The phospholipid itself is a very unique molecule in that it's an amphipathic molecule, meaning that there's a hydrophilic head and a hydrophilic tail region, and they actually form this bilayer like this. So if my hydrophobic head, if I use these little circles to illustrate that, and the little tails are the hydrophobic tails, they're little hydrocarbon tails. The membrane is very fluid, much like almost like a vegetable oil, so very fluid. There's other stuff in here, such as cholesterol and protein molecules and that, but the primary makeup of that membrane is the phospholipid bilayer. So the inside of the membrane is a hydrophobic region consisting of your hydrocarbon tails, and your head region is a glycerol, 3-carbon glycerol molecule attached to 2 of those carbons are your fatty acid tails, and then a little phosphate group. And that gives it a little polarity and makes it more hydrophilic, water-loving. And so it's a unique composition of that plasma membrane, and it serves as a barrier to the external environment for that cell. Inside the cell, if we start with the nucleus. Now the nucleus itself is bound by 2 membranes. There's an inner membrane and an outer membrane associated with the nucleus, but they're very large pores called nuclear pores or channels that allow stuff to move in and out of that cell. And we've got to move big molecules in and out of a nucleus like this, such as RNA or nucleotides or enzymes, different things like that that can move in and out of that nucleus. Contained inside that nucleus would be the chromatin, which would be all the DNA associated with this particular organism. It only becomes discrete chromosomes when the cell is fixing to divide. So normally it's more just a tangled mass of DNA, although there's order going on there, but we'll call it chromatin. Chromatin also has associated with it all kinds of different proteins, structural proteins, enzymatic proteins, regulatory proteins. There's all kinds of different proteins associated with that DNA in that nucleus. One of the primary protective proteins is called the histones. The histones is a large class of proteins. There's different types of histones, but they're the packaging proteins that help to protect and coil the DNA up when it's coiling up into chromosomes. Also associated with it, and you can see here looks like a center of the eye or something like that, is the nucleolus. The nucleolus found in the nucleus is the site of rRNA or ribosomal RNA. Ribosomal RNA makes up the ribosomes out in the cytoplasm, and that's the sites of protein synthesis associated with a cell like this. So we have our nucleus. We'll call it, textbook term here, command center of the cell. That's where the genetic material is contained for the cell. Not the only place. We can also find DNA in a cell like this within the mitochondria as well, as mentioned earlier. So extending out from this nuclear membrane is the endomembrane system. And the endomembrane system consists of the endoplasmic reticulum, ER, rough and smooth. The rough ER is associated with ribosomes that will attach to that endoplasmic reticulum. Sometimes a ribosome is literally told, I'm personifying here, told by messenger RNA that's produced in the nucleus, messenger RNA that's produced when a gene is being expressed. That messenger RNA is shuttled out into the cytoplasm, and depending on the sequence of nucleotides within that messenger RNA, which is literally templated off a strand of DNA, coding DNA that serves as a gene. So you have messenger RNA that's produced in the nucleus, shuttled out to the cytoplasm where it's read by a ribosome. If there's a certain sequence of nucleotides in that stretch of RNA, that messenger RNA, that ribosome will attach to the messenger RNA and shuttle it to the endoplasmic reticulum, if that protein or that polypeptide is to be deposited into the endoplasmic reticulum. So the ribosomes that are attached to the rough ER are only attached temporarily, but there's always some protein synthesis going on, so there is a certain region of that endoplasmic reticulum that will be studied or associated with ribosomes. There's another portion of the endoplasmic reticulum that tends to lack ribosomes, and we call that the smooth ER, smooth endoplasmic reticulum. Now, it may contain proteins that are being shuttled through that endoplasmic reticulum tubules, where those proteins can be packaged and then secreted out of the endoplasmic reticulum as little vesicles that can go somewhere else in the cell. Smooth endoplasmic reticulum can also be in the process of producing more membrane that can migrate to the plasma membrane or to another organelle. So the smooth endoplasmic reticulum can be involved in packaging proteins or in the production of various lipids, especially phospholipids. Another part or extension of the endoplasmic or the endomembrane system would be the Golgi body or Golgi apparatus. It can receive vesicles from the endoplasmic reticulum. If the vessel contains proteins, oftentimes those proteins are further modified inside the Golgi apparatus, or maybe a carbohydrate chain or chains are attached to that protein, or a lipid is attached to the protein, or more modification takes place with the protein molecule or even lipid molecule. So further modifications of various molecules takes place in that Golgi apparatus, and it essentially is an extension of the endomembrane system. So your endomembrane system consists of a few players. Certainly the endoplasmic reticulum that's involved in modifying proteins if they're deposited in there, but also involved in the manufacturing of certain lipid molecules. So endoplasmic reticulum, both rough and smooth, part of the endomembrane system, but also the Golgi apparatus, which is not attached to, but associated with, just through the movement of vesicles being shuttled to it. And a vesicle is nothing more than a sphere surrounded by phospholipids. And so those vesicles can shuttle stuff around, substances around in the cell. And so vesicles can bud off of the smooth ER to the Golgi apparatus with compounds in there for those compounds to be modified in the Golgi apparatus. Then vesicles can either come from the Golgi apparatus or even the smooth ER to other vesicle-like organelles in the cell. There's a couple of vesicle-like organelles in a cell like this, and then they would be the lysosomes. Lysosomes function as digestive organelles in eukaryotic cells. Lysosomes contain digestive enzymes and tend to be very low in pH. They act like little stomachs or little digestive systems for a cell like this, and they are products of that endomembrane system. So you have vesicle-like organelles like that. In a plant cell like this, there are also peroxisomes. And peroxisomes are in the business of breaking down fatty acids. And peroxisomes contain a particular enzyme called catalase that can break down hydrogen peroxide that is oftentimes used to help to break down lipid molecules. But anyways, a peroxisome is another type of vesicle-like organelle involved in digestion. Also in the cytoplasm here, we can see the little mitochondria. Mitochondria is an organelle associated with the production of ATP. Sometimes it's defined as the powerhouse of a cell, right? So we think about cellular respiration as associated with those kinds of organelles, the mitochondria. In a plant cell like this, we have not only mitochondria, but we also have chloroplasts. So you have the site of cellular respiration, the mitochondria, and the chloroplasts are the site of photosynthesis in a eukaryotic plant cell or even eukaryotic algae cells. So those are two energy-deriving organelles, the chloroplast, photosynthesis, and the mitochondria, cellular respiration. Also associated with these cells are microtubule organizing centers. Now, something that's common with eukaryotic cells that we don't see in prokaryotic cells is an internal structure in these cells called the cytoskeleton, which is made up of various types of protein molecules. There are three basic types of cytoskeleton protein molecules. One is actin filaments that are relatively thin, little small filaments, usually found around the perimeter of the membrane and help to support the cell, can help the cell move by formation of pseudopods if it's a cell that does that, can help the cytoplasm move around in a process known as cytoplasmic streaming. Intermediate filaments can act as scaffolding through the cell to help just give it rigidity and structure. And then microtubules are used in a variety of functions. Microtubules can form flagella if the cell has a flagella or flagellum. Microtubules can also be associated with moving actual organelles around in the cell, especially in conjunction with motor protein molecules that can attach to organelles and shuttle them along a microtubule filament. Microtubules are associated with the formation of spindle fibers when cells like this are undergoing nuclear division, such as mitosis or meiosis. So microtubules are certainly large, and they're the workhorse molecules within cells like this. So we find these microtubule organizing centers close to the nuclei in these two cells. A little different in an animal cell. The microtubule organizing center is highly concentrated in a region called the centrosome. And you can see here there's a pair of centrioles, and these are just little microtubules that are already kind of linked together. And if this cell was to undergo cell division, the centrioles would divide, and we give it a whole new term, the asters would form and all that kind of stuff. But at any rate, you do usually have this microtubule organizing center, usually buffered up right in against the nucleus of a cell like this. Couple of distinguishing differences between a plant and animal cell. Plant cells produce an external cell wall made of cellulose, and that helps to protect this plant cell from the outside environment, but also helps to protect it from itself. This contains that cell because the pressure inside a plant cell is usually high because the tonicity, the solution